Okay, so for this chapter, we have already covered the first two objectives on our handout, which was the components of DNA. And then we looked at the central dogma of molecular biology. And now what we're going to do is going to look how we zoom in on some of these things and we use the tools that are already available within biology, and we manipulate those in order to ask questions about many different types of processes. And so what I have created is, here's your list of things, and you can go in and you can write down these definitions. I have links to the different videos, or not, well, some different videos that you will have watched for this particular chapter. And then there's also a links to a different way of defining them. So if the videos work for you, that's probably all you need. Or if it helps you to have a description of those things, there are links within the PowerPoint. And then what we will do during class is I'll have like things you can work at, and like a card game that you'll sort all the different things to test yourself on those as well. So one thing we have already talked about is that cDNA. And then we have this really long list of things that may or may not be familiar to you yet. Then we have PCR, which is a really important one. You're going to be doing PCR in lab. And there's a specific video for that, the hybridization process, and then oligonucleotides. So some of these things are individual components, and then other things are experimental procedures. So when you go to the full PowerPoint and you find the slide for this one, all of these links are going to have all of these defined for you. So your job right now is, I could go and I could tell you all the definitions, but honestly, all the definitions are already written right here. So what I want you to do is watch the videos that I have available, and then see if you can figure out which of these definitions goes to which component or procedure. And then during class, we will test to see how far you got, and where we might need to fill in some gaps between that, which we will do that exercise during class, and we'll answer questions for those during class as well. So you have these for your resources, and I also have some images that might help you as you're going through. Like, you can look at this and say, okay, well, what does this mean? How does this compare? These are some of the images that you can think about. And so if this helps you process it, again, we'll talk about it in class as well. But there are a few extra images in there. And if you wanted to, you could kind of pre-practice, because I have a link to the cards using class. And these are going to be the things, these are going to be the definitions. And then I also have specific images that are going to go along with those as well. If you process things better by seeing a picture, I will have the name, the definition, and a picture of like how we use it, or what the actual component is going to look like. So spend a little bit of time going through and clicking on these different ones to see how those resources are going to help you understand each of these components or experiments.